SAMERA stands for Semi-Automated Multi-Endpoint ROS Activity Analyzer. So first, I want to raise a question. Why do we concern about the ROS activity of PM? We have already used the PM 2.5 concentration as the health metric. However, from our previous studies, people have found that different PM are composite with different PM components. For example, we know 1 gram of sulfate salt could not be equal to 1 gram of iron because they have totally different property and they show very different health metric. However, they are equal in the mass. This is not fair for different sites. Here, we want to explore a new health metric called oxidative potential. This oxidative potential actually reveals the ROS, reactive oxygen species production ability of PM, and therefore it can give us a better impression whether the PM we are investigating could be more or less toxic. The system SAMERA consists of 3 parts. Since it is an automated system, here we use these 3 pumps to realize the automation of the system. You can see these long tubes like spiders' legs. They reached out to the different chemicals and also our samples, finally to the incubation vials. This system is controlled by a software called as clone control. The clone control draws solution from different chemicals and dispenses them into the incubator. And here the incubator as the second part lead to the reaction. After a certain time point, a small fraction will be drawn out into our LWCC and a flow cell here in the HORIBA spectrofluorimeter. These 2 parts consist of the third part of the SAMERA system, which serves for the measurement of both absorbance and fluorescence. Based on the absorbance and fluorescence change, we can determine the concentration change of either ROS or the antioxidants. SAMERA system uses 3 different antioxidant consumption rate and 2 OH generation rate as our endpoints for measuring oxidative potential. Since there is no consensus which oxidative potential is the best to represent the health metric, we adopted all these 5 endpoints, including OPAA, OPGSH, OPOHSRF, OPDCT, as well as OPOHDTT in our system. This system could measure a single sample oxidative potential in all 5 endpoints within only 3 hours. This time reduction is a huge improvement compared to previous studies using manual operation. Therefore, our hands could be released from those dumb experiments, and we can use more time to do the data analysis. SAMERA is my proud work, and this is designed and realized by me in my past 2 years. I am experienced into this automation of the system as well as the analysis of chemical and oxidative potential. Also, I am adept to the data analysis on the spatial temporal variability of Midwest US, which has served as my topic of my PhD thesis. I also have a presentation on this Friday, which is on the same topic. I welcome all of you to attend my presentation, and I will be I will be happy to answer any questions on either the system or my study. Please contact my email, which has been attached to the poster. Thank you so much.